So one of the ways that you can use the Hardy-Weinberg equation is to apply it to a real life situation. You would do this if you want to understand what genotypes you have in your population. Maybe you're trying to figure out how likely it is that somebody is going to be homozygous recessive for a particular disorder. Or maybe you're trying to figure out how many heterozygote carriers you have for a particular disorder. So let's use a real life example of this. So remember, Hardy-Weinberg is used to determine whether or not evolution is occurring in a population. So we'll talk about how the Hardy-Weinberg conditions are met in this particular example. So in this example, we're going to use Hardy-Weinberg to estimate the percent of the population who are carriers or heterozygotes for an inherited disease. That disease is called PKU, or Phenylketonuria, and this is a metabolic disorder where individuals have the inability to break down phenylalanine, to metabolize phenylalanine, which is an amino acid. So this is pretty rare, 1 in 10,000 births. You may or may not have ever heard of this. It is something that is very survivable. You avoid having large quantities in your diet by making sure that the foods you eat don't contain phenylalanine or don't contain a lot of phenylalanine. So we're going to assume that this allele is in Hardy-Weinberg equilibrium. So let's talk about what that means for a real population. So this is in a little bit different order than our list previously, but that doesn't really matter. So first condition, there are no new mutations for PKU. So this means that the allele in this PKU gene doesn't have any mutations in it. And we can assume this because mutation rates are low. So not only are mutation rates low, but there's even less of a chance that it's occurring in this particular gene that we're talking about. Random mating. So people don't base their mating, their choosing of a mate, choosing to have children with somebody based on this allele. It's probably not at the top of your list for dating criteria. So people are not being selected against because they have PKU. And also inbreeding is not common in the United States, so we can assume that that is also not happening, that people are not being forced to mate within a particular subset of the population. There's no differential survival. So historically, maybe there was. If you had a diet that was high in phenylalanine and you didn't realize that you had this, you might have there might be some selection against individuals with this disorder. But we can assume that nobody is surviving at a lower rate because of this disease, because it can be controlled with dietary restrictions. So this is sort of a special case. This is a case where there might actually be selection against this disorder, but in this particular case, because it is controlled by diet or maybe in another disease by medication, there's no one is surviving any better because they do or do not have PKU. No genetic drift or large population size. We'll just assume that the population size in the United States is large enough to withstand any changes due to chance. And then no gene flow. So this one is a little bit harder to think about because if you think about it, there are people moving in and out of the U.S. population at all times. But gene flow, if the population where somebody is coming from has an allele frequency that's the same for this allele, then it's not going to change the allele frequencies. So the gene frequencies in other populations are similar, and our population size is large enough that gene flow is not going to affect the allele frequency of this allele. So we can assume that the PKU allele is in Hardy-Weinberg equilibrium. I'm sorry if there was a little blip in the recording. I had to stop and go back because I made the mistake that I was about to tell you not to make when I was talking. So if there was a little blip, just ignore it. We'll continue. All right, so we are going to use the information that 1 in 10,000 individuals has this disorder in order to calculate our allele frequency. So remember, if all of our Hardy-Weinberg conditions are met, we can use this Hardy-Weinberg equation. So the frequency of homozygous alleles is going to be equal to the frequency of homozygous individuals is going to be equal to the frequency in the population. Because remember, Q squared is a genotype frequency, and the individuals who have the recessive allele are going to be the that have the recessive trait are going to be the homozygous recessive individuals. So the number one problem that students make in this situation is that they're given a frequency. They are told that 1 in 10,000 individuals have the disease. When it is a recessive trait, that is the genotype frequency. But the number one mistake that students make is they set that frequency, the 1 in 10,000, or the number of individuals with the homozygous recessive phenotype, equal to the allele frequency. And you cannot do that because remember, individuals have 2 copies of each allele, but also there are alleles in the heterozygote individual. So you have to calculate the allele frequency. When you are given a phenotype, such as the individuals with PKU disorder, you are given a genotype frequency. The number of recessive individuals in a population is equal to Q squared. So we can use that information to calculate Q. So Q squared is equal to 1 in 10,000, or 0.001. Okay? If we then square root it, we find that Q is equal to 0.01. We now have an allele frequency. We started with a genotype frequency, and now we have an allele frequency. This is really important, and it's the number one place where students mess up this problem. So we were given a genotype frequency, the number of individuals with PKU. We use this to calculate the allele frequency, to calculate our Q value. We cannot use the opposite, the 9,999 individuals to calculate the p-value, and I'll talk about why in just a minute. Instead, we're going to use the idea that P plus Q equals 1, therefore P equals 1 minus 0.01, and therefore the p-value, or the frequency of the dominant allele, the normal allele, is 0.99. So, from there, our problem asked us to calculate the number of heterozygous carriers. Well, now we have calculated we've calculated P and we've calculated Q. So now that we know P and Q, we can just plug this back into the right part of our Hardy-Weinberg equation. Remember that the genotype frequency of heterozygotes is equal to 2pq, so we just plug it in and calculate it. 2 times the frequency of p, which is 0.99, times the frequency of q, which is 0.01. Means that the frequency of heterozygotes in a population, or the number of carriers for this disorder, is 0.0198, or roughly 2% of the population in this situation are carriers. Let me go back just a little bit and talk about why we cannot calculate P directly. Now, if you remember, I said that if you are a heterozygous individual, you have the same same phenotype as homozygous dominant individuals, but you have a hidden Q allele. And because our population of normal individuals consists of individuals who are homozygous dominant and individuals who have a hidden Q allele, you cannot use the number of dominant phenotype individuals to calculate P. Because there's a small number of Q alleles hidden in that population. Now you can use it the other way around. You can use Q squared to calculate the population because you're using a set of individuals whose phenotype you can see and whose genotype you know. You know all of the individuals who have that disorder are recessive, and that's why you can calculate Q from your Q squared. So if you do not understand what's going on here, make sure that you ask in class, because this is where students lose points, and it is fundamental to the understanding of those allele frequencies and genotype frequencies to be able to get this problem correct.